Today I'll be talking about my core project during my postdoctoral years, which is entitled Predicting mRNA Abundance Directly from Genomic Sequence Using Deep Convolutional Neural Networks. So one simple observation, if you sequence any tissue of your interest in its RNA landscape, in bulk or in single cell, you often get a readout that looks like this, where on the x-axis, we're visualizing just mRNA abundances. And if you just create a histogram out of that, you see something very simple that you've probably seen many times in your past, which is that gene expression levels from gene to gene vary by many orders of magnitude. In this case, log base 10, we can frequently see they vary by 5 to 6 orders of magnitude. And that simple observation is something that I've always wondered about. I've always wondered, why is it that this gene in the genome is expressed 5 or 6 orders of magnitude more than another gene? And when I began this project in 2015 or so, there had been a little bit studied about just gene regulation as a whole in the cell. So if we think about this as a contribution to the levels of the proteome or levels of the proteins in the cell, there are some interesting studies from Europe that actually specifically interrogated the rates of transcription, translation, as well as degradation rates for mRNAs and proteins. And one of their key findings, later reanalysis of this data set from UC Berkeley, uncovered that mRNA steady state levels contribute about 84% of this piece of the pie, whereas there's a relatively modest contribution of protein degradation and translation rates. And that really suggests that whatever's going on, the transcription is the biggest piece of this pie with a smaller contribution of mRNA degradation. But not that much is known about what is governing the levels of mRNA. And that was kind of the basis of my interest in this question. So if we think about the different modes of gene regulation holistically from the perspective of what governs mRNA steady state levels, we can think of this as two sides of the equation. So different determinants that govern the birth rate of mRNA and other determinants that govern the death rate. With respect to the birth rate, of course, there's the promoter sequence at the beginning of a gene that governs maybe the basal activity of transcription, but there are also distal elements such as enhancers, which can be up to a megabase away. And these are often associated with histone marks such as H3K27 acetylation. But this process of gene activation is counterbalanced by processes of gene silencing, often associated with polycomb target regions or marked with H3K27 trimethylation. So somehow all of these different forces govern the transcriptional output of a gene. And as that mRNA is then transported from the nucleus to the cytosol, then different determinants impact the death rate of mRNA, such as the presence of microRNA binding sites or AU-rich elements or familial factor binding sites. Determinants such as coding exon density are also associated with mRNA decay. And so when I was interested in trying to model something like this, I tried to do a deep dive of literature at that time period. And just one of the things that I saw is that there's some algorithms that try to directly predict mRNA levels directly from genomic sequence using sequence-based features such as transcription factor binding sites. But when I began this project, most of the techniques that attempt this problem simply weren't that great at the time. So they explain around 20% of the variability in gene expression levels. And in contrast, there are other techniques. For example, you can use a massively parallel reporter assay to measure promoter activity in parallel for thousands of promoters. And that's kind of another mechanism by which one can train a model or simply look at the relationship to a sequence of interest. Alternatively, a big consortium in America was the ENCODE project, and now they provided data for hundreds of different transcription factors with respect to performing chromatin immunoprecipitation and sequencing of regions that the proteins are bound to. There's also ChIP-seq of histone marks, such as H3K acetylation, trimethylation, or H3K4 mono or trimethylation. All of these were used later on to develop quantitative models to predict expression levels from histone marks or ChIP-seq datasets, and DNase I hypersensitivity measurements, which measure open chromatin. And so my argument is that while the first one is kind of an ideal goal, the techniques weren't very good, but it's nice that these other models did actually pretty well in predicting gene expression levels. However, there's not a direct causal interpretation in building a quantitative model like that. So you might simply detect associations between the enrichment of these peaks at promoters and gene expression levels. But it kind of leads to this chicken and the egg question, which is, was expression governed first and lead to the deposition of these marks, or were the marks deposited and did it lead to the gene expression program? So that's kind of like a difficult problem to dissect in the epigenetics field. So I was kind of interested in taking a deeper dive into the first group of approaches and see if I can make any headway into that problem. So the three fundamental questions I was interested in asking in my project was, how far can we get in predicting mRNA steady state levels in a cell using nothing more than genomic sequence? So just from a theoretical perspective, is this problem even feasible, and what is achievable in this question? The second question I was interested in is simply, how is information dispersed in the genome? What is the relative contribution of promoters, mRNA half-life features, enhancers, or other elements? Is it the case that information is highly centralized at the promoter, or is it largely distributed across distal elements? And the third question I wanted to ask is, using these models, what can we learn about gene regulatory mechanisms operating in different cell types? So the analogy that I'll give to the approach I took an approach based upon the use of convolutional neural networks. And the analogy I'll give is that of cognition. So if we think of the brains of different species, each of them is organized in a different hierarchical fashion. And that to some degree governs what each organism can learn about its environment and how it can process the input of the stimulus in the environment and lead to some degree of abstraction of that stimulus to create some notion of what the environment is. So similarly, that's to some degree, there's some analogies to what neural networks accomplish. So their goal is to feed information from an input space, learn hierarchical principles and abstractions, and ultimately govern a prediction, which is the output of the network. In this case, a lot of this field emerged from the field of computer vision. So as input, you can see this pixelated image, for example, of the digit 5. And the task of the network is to learn a set of parameters across many training examples to finally classify a digit. And in the middle are what are called data convolutions, which basically are submatrices that scan this image. And if you can hierarchically order these, they can extract more and more abstract features, such as how curves and lines are put together to create this digit. So what I want to do is to try to repurpose these types of networks to the task of predicting steady state mRNA levels. And so instead of this pixelated image, now we have an image, quote unquote, which kind of looks like a DNA sequence. So we have a DNA sequence, which you can one-hot encode into this matrix. So for each column, you have the binary vector, is it an A, T, G, or C. And here I'm just showing you the transcriptional start site plus or minus 4 kilobases as my input. I can feed this again through several series of convolutional layers and try to combine these with sequence encoded features governing mRNA half-life. And my final output is a prediction of gene expression level. But is it the case that 4 KB is optimal? I'm not sure. So maybe it's this other alternative architecture is better. One should use maybe 1 kilobase instead of 4 kilobases, or maybe instead of 2 convolutional layers, we should use one convolutional layer. All of those are basically different architectures, and each of these architectures is specified by a group of hyperparameters. And as I was mentioning, the structure can govern the types of mathematical functions that these architectures can learn. But it's unclear what the right one is. So the approach that I took was to try to, as input, take these two types of features, the promoter sequence. I tried to take an extended region, plus minus 10 kb, and try to let the network learn what the optimal region is, as well as alternative set of features governing mRNA decay. So I performed a hyperparameter search to look across many different types of architectures to discover a solution that could learn this principle the best. I used two algorithms. One is called the tree of Parson estimators, and the second is called simulated annealing. And on the x-axis, what I'm showing is the number of iterations. A single iteration alludes to one particular architecture that I'm searching. And on the y-axis, I'm showing the performance on a validation set. So a lower number is better because we're decreasing the mean squared error of this problem. So what I found is that as I search along the x-axis, I'm basically plotting here the very best model that I've discovered so far. And I'm searching hundreds and hundreds and hundreds of different architectures, which is a computationally intensive procedure, often trained on GPUs. And ultimately, I converge and plateau at an architecture that does the very best, using this approach in red, the tree of Parson estimators approach. And what I ultimately discovered is that this is the architecture that was ultimately learned. It's not necessarily the optimal architecture, but it's a pretty good architecture that's perhaps suboptimal, but it captures much of the variability in the data set. So the architecture that was learned ultimately used 7 kilobases upstream of the transcriptional start site and 3.5 kilobases downstream. Again, and then it fed this into a series of 3 convolutional and max pooling layers. Max pooling will reduce the dimensionality of this problem. And it finally feeds it into 2 fully connected layers, one of which I've integrated these mRNA half-life features. Now you might wonder, what are these mRNA half-life features that I'm using in this problem? And basically, I used to work in the lab at MIT called the Bartel Lab. And one of their projects was to try to interrogate what sequence encoded determinants govern mRNA half-life. And a paper by my former colleague, Noah Spies, discovered that there's several simple features that can explain a good degree of mRNA half-life, including coding exon density, which alludes to the number of exon junctions per unit length of the coding sequence of the mRNA. That's actually the dominant feature that governs, or is at least correlated with mRNA half-life. But in addition, there's smaller determinants that can be easily computable from a genomic sequence, such as the GC content of the 5' UTR, open reading frame, and 3' UTR, as well as simply the length of each of these different regions. And these are integrated as other forms of input that can help boost the performance of this network. And the final prediction here, as I'm showing on the right side, is an output, which is the log of the mRNA levels. So how does this model perform? I looked at a set of 1,000 genes in my test set that had never been before seen by my method. And on the x-axis, I'm plotting the predicted expression level from the neural network. And on the y-axis, I'm predicting the median expression level in a number of different cell types in human cells. And what I found over many rounds of iteration and optimization is that I could achieve finally an R² of around 0.59 or so, almost 0.6 in the human. I also performed this in the mouse, and I detected an even higher R² of around 0.71 or so. So overall, using something just including promoters and simple sequence-based features governing mRNA half-life can predict a quite a substantial amount of variability in gene expression levels. I wanted to extend this to individual cell types. So I generated cross-validated predictions for K562, which is a hematopoietic cell line. And now for an individual cell type, the predictions are slightly worse. So instead of 0.59, it's 0.51 of an R squared. And I wanted to try to understand what can we actually learn from what the model is failing at. So one of the things that I investigated is I looked at the outliers of this plot. And from the outliers, I discovered something kind of interesting, which is that many of these outliers basically all were related to, in this particular cell type, subunits of the hemoglobin pathway. So why this is a particularly interesting finding is that in the 1980s, when enhancers were first articulated and discovered, these were kind of some of the first loci in the human genome discovered to be very highly activated by strong enhancers. And so I wanted to try to exploit that information by explicitly looking at the errors of my predictions. And so these errors are basically just residuals, and it's simply the difference between the observed and predicted value. And I'm plotting those errors here on the x-axis, that residual expression level. And I plotted that for a group of all of the genes in the genome. So shown in black are most genes, about 10,000 of them. But shown in red are a subset of genes associated with H3K27 acetylation marks. These are regions that were annotated to be associated with stretch enhancers in K562 cells specifically. And what I found was that my model was consistently underpredicting these genes, in that presumably the stretch enhancers are activating gene expression more than the model might predict given the promoter sequence. In contrast, I also observed that the model was over-predicting genes that were associated with H3K27 trimethylation domains, which is a classic mark of gene silencing. So we could kind of detect a cohort of genes that were both activated, presumably by enhancers, or silenced by these repressed domains. The same pattern kind of bore out in a different cell type. Here I'm showing mouse embryonic stem cells. And in this case, group work by Rick Young at MIT has also annotated throughout the cell type super enhancers that are regions of very large H3K27 acetylation domains and other marks associated with mediator which activate transcription. His group also annotated regions associated with polycomb targeted regions. And we could see kind of the same pattern of activation or repression of these relative to the model predictions. Now I just would turn our attention briefly to microRNAs, which is a topic that I used to study quite a bit in my PhD. And microRNAs are small molecules that target messenger RNA, and they arise from hairpins in the genome. They're processed by 2 enzymatic steps into a mature form, and they're integrated into a protein called argonaute. And argonaute then in the cytoplasm can target particular messenger RNAs through a region called the seed region. These are just regions of Watson-Crick complementarity between the microRNA shown in red and the target mRNA, frequently through motifs located in the 3' UTR of messenger RNAs. And the ultimate consequence of this pathway is that the microRNA binding to these different elements leads to a small degree translational repression of the target, but to a much higher degree direct degradation of the messenger RNA. So what I was interested in is investigating whether these residuals can actually give insight into active microRNAs in different cell types. So one cell type that I interrogated was mouse embryonic stem cells again. And I did something very simple, which is there's small RNA sequencing data from the cell type. And I simply looked at a pie chart. I generated a pie chart which basically looks at the abundance of different microRNA families as defined by microRNAs that share this core seed region that I was mentioning. And I asked the question, if let's say hypothetically, I knew literally nothing about the microRNAs active in the cell type, is it possible to try to infer or reconstruct something about active microRNAs in the cell type? So I took my residuals and then looked at the Spearman correlation between a predictive algorithm called target scan, which takes each microRNA in the genome and attempts to predict the level of repression that targets will exhibit. And again, that's purely based on the sequence of 3' UTRs of these genes. And I looked at the Spearman correlation between these predictions as well as the residuals from my work. And I found a group of microRNAs that had the very highest Spearman correlation. It turned out that these microRNAs that had the highest correlation of them all corresponded to the most abundant families in mouse embryonic stem cells. And we could see that for the greatest outliers of the Spearman correlation and histogram distribution.
Sorry, so this is for the residuals of the model that's trained on average gene expression across all tissues.
Yes, but in this particular case, I'm also training a model on embryonic stem cells. That's a cross-validated model. And then I'm fitting that and extracting the residuals in mouse embryonic stem cells.
Okay. So it trains on the average expression, tests on embryonic stem cells, and then...
Yes, exactly. I can do it either way. I could train on the median and then extract the residuals on an individual cell type, and then look at the correlations to these predictions. Or if there is data from the cell type, I can train on the specific cell type and then look at the residuals again based on cross-validated predictions.
Okay.
So basically the conclusion of the slide is simply that it is possible to glean insight into active microRNA families from a gene expression profile alone using this technique. I wanted to assess what else is possible with a type of model such as this one. So one of the things I was interested in is just looking across an entire genomic locus. So here I'm showing about 600 to 800 kilobases of chromosome 1 with all these different genes in the middle. And plotted below that is all the CpG islands that have been annotated in this segment of the genome, as well as states of chromatin as annotated by a hidden Markov model called chromHMM in 2 different cell types. And the chromHMM tracks show these different regions in red, which are basically its predicted promoter regions. And below that, I'm plotting the total counts as assessed by CAGE, or CAP analysis of gene expression, which basically measures transcriptional activity from the 5' end of a transcript. And you can see that the CpG islands are aligning quite well with these predicted promoters, and those in turn align pretty well with regions of these peaks of CAGE activity. But in addition, I ran my algorithm on a sliding window across this entire region at 100 nucleotide resolution. And one of the things that I discovered is that the predicted transcriptional activity by my algorithm pretty well mirrors the CAGE activity or transcriptional activity of this whole region. So we can actually use methods such as this one to create a continuous prediction across large genomic regions and the whole genome if one wishes. So one of the complex things about neural networks is that they're frequently criticized for being black box techniques and that they're very difficult to interrogate or interpret. And despite that being somewhat true, there are some techniques in the field that have attempted to actually interrogate and understand what the network has learned. One of these techniques called integrated gradients, basically for each prediction, it goes back in the input space and tries to assess what specific regions of the input were highly relevant or led to the high prediction of this particular input. So what I'm showing here on the x-axis is the position relative to the transcriptional start site, which is kind of the spatial coordinate that relative to my input sequence. And then I'm binning genes into one of three categories: low, medium, or highly expressed genes. And on the y-axis, I'm showing this readout called the saliency score, which basically computes the original signal that contributes most to the prediction. And what I found using this technique, averaging across thousands of genes for each of these bins now, is that the algorithm actually very lowly weights most of the space. It only actually very highly weights a very small region around the core promoter centered around 0 here, which is the region of 0 plus or minus around 500 nucleotides. And all the other regions outside of that, it weights to a small degree, but in a way that's not that highly useful to the model. So the nice thing about this is that without any knowledge whatsoever of what a transcriptional start site is or what a promoter is, quantitatively the model can learn something about the size of a promoter, in the sense that something that is predictive of transcriptional abundances. And the same pattern bore out in both human and mouse together. What else did the model learn? So I was very interested in learning what are the sequence encoded features in the promoter now that the model has learned. So my approach was to do something as simple as possible, which is just generate predictions for a group of genes in the test set. And then basically I noticed that you could fit this into a bimodal distribution into two classes: a subset of genes that are very lowly expressed and lowly predicted, and a subset of genes that are highly predicted. So I fit two bimodal distributions on this distribution, and I just simply asked the question, what sequence-based features discriminate these two classes, using an algorithm called DREAM, which is part of the meme suite. And after doing that, I discovered an extremely simple feature. And that simple feature was common to both humans and mouse. That feature was the presence of CpG dinucleotides. And it turns out that the density of these dinucleotides, this model predicted to be highly associated to gene expression abundances. So now at this stage, the model has made a very specific prediction, which is that the density of CpG dinucleotides at mammalian promoters should be highly associated to gene expression level. So I tested that directly by empirically looking at the expected to observed frequencies of dinucleotides in different classes of genes and their abundances. So here I'm splitting up the genes in the genome into 4 classes: a cohort of genes that are largely non-expressed, and genes expressed at a low, medium, or high level. And what I'm plotting on the y-axis here is the observed relative to the expected ratio of CpG dinucleotides in different registers relative to the transcriptional start site centered at 0, plus or minus 2,000 nucleotides. The way the expected frequency is calculated is simply by looking at the frequency of C's and G's and multiplying the two. So the joint probability of C and G co-occurring feeds into this expected probability just by chance expectation. What would you expect? And the specific prediction of this model did bear out in that one could see that there is this relationship that promoters that are more highly associated with this dinucleotide do seem to be more highly expressed. So this was all work that I discovered purely computationally, but it was very nice to see that there were several other studies that came out in around the same time period that I was doing this work that were completely orthogonal experimental assays to experimentally interrogate this problem. So the first was a paper by Bas van Steensel's group in the Netherlands, and his group used massively parallel reporter assays to fragment the genome and measure the promoter activity of all the regions in the genome. And one of their figures showed that if you plot this expected CpG density to the observed density, sure enough, those promoters that fall along the diagonal where the observed and expected meet, these are regions that show the highest amount of promoter activity, implying that there's a causal relationship between CpG density and gene expression level. This question was also interrogated by a different group in Switzerland, which actually, again, used reporter assays to interrogate how the presence of CpG dinucleotides enhance promoter activity. In their case, they found a correlation, and they went in and then deleted or mutated some of these CpGs and found that this diminished the correlation in the relationship between the density and promoter activity. So all three of these different studies are taking different approaches, but all kind of converged on the same finding that the nucleotide density of CpGs governs to some degree gene expression levels. And I just want to briefly mention that CpG dinucleotides have been observed in mammalian promoters for decades, since like the 1980s or so. But as far as from my reading of the literature, it was not appreciated until recently that their density might actually directly govern or be related to transcriptional abundance. What I want to do finally, this is just my last result now, I wanted to compare the performance of my method to a number of other methods attempting this problem. Shown in purple are the subset of techniques that use only genomic sequence in this gene expression prediction task. And shown on the bottom here are 5 different types of information that different algorithms use. I draw a plus here depending on which of these 5 different approaches, different types of input are used. And the first thing I did is I just looked at the performance of my technique called Espresso relative to alternative techniques that use only sequence. I established some simple baselines in this paper as well, but the existing publications that attempted this problem had about 1/3 to 1/4 of the predictive performance as my implementation. But in addition, if we look at these techniques that use, in some cases, hundreds or thousands of different biochemical data sets to attempt this problem of gene expression prediction, we can see that in most cases, my algorithm do better than some of these techniques. And in other cases, it does similarly to techniques that use a vast number of experimental inputs to this prediction problem. So finally, I just want to mention that I have a very strong open source and open access philosophy, and I developed this software as an open source tool, and it can easily be used and deployed using this associated website. And all the figures of our paper I've implemented in this GitHub repository to make it fully reproducible. Finally, just in conclusion, I've hopefully demonstrated to you that these deep learning based methods are capable of predicting about 50 to 70% of the variability in gene expression levels while only using genome sequence encoded features. The prediction errors can actually give insight into different gene regulatory mechanisms active in a cell type, including the activity of enhancers and silenced heterochromatin domains. The model is capable of automatically learning some biologically relevant functional regions, such as defining quantitatively the region of a promoter. And it makes a specific prediction as to the relationship between CpG density and in the core promoter and gene expression levels. This sequence-only model is competitive with models that use far more experimental data as well.